Anything else about osmosis? What's the other way you could explain it besides water is moving down an osmotic gradient?
Towards the solute.
You could say water is moving down its concentration gradient. Because where you have the lower solute concentration, you have a higher water concentration. So water truly is moving down its own concentration gradient. I just tend to look at it from the perspective of the solutes. Water is going to the area of higher solute concentration, higher osmolar. Filtration. What's the driving force? Hydrostatic pressure. What is hydrostatic pressure? It's a fluid pressure exerted on the walls of its container, which could be a cell, a vessel, a beaker. Where does filtration occur in the body? I'm not looking for an organ. Much simpler than that, because it happens all over your body, not just in the kidneys. That's just one example. It happens everywhere. Cross capillary walls. Filtration occurs in one place only, across capillary walls, including in the kidneys. So yesterday when we were looking at the tissue slides for the different types of epithelium, we saw one slide that had 4 pictures for simple squamous, and I had arrows because there was a lot of other cell types and tissue types in there, and I wanted you to see exactly where the simple squamous was. That first top left image was in the kidneys, Bowman's capsule, and inside a ball of capillaries called the glomerulus. So still in the end, yeah, in the kidneys, but it's ultimately across those capillary walls in the kidneys. Now the fluid that gets pushed across the capillary wall, what do you call that? Once it ends up on the other side. Filtrate. So when the fluid is in the capillary, before it moves across the wall, what's it called? The fluid component of blood. Plasma. As soon as it gets pushed across the capillary wall, well, let me do it this way. We now call it interstitial fluid, but it is the filtrate. So calling it filtrate is just saying that this fluid came from the other side of that barrier. This is the stuff that got pushed across. It made it across. Everything that made it across is the filtrate. But now in the new environment, it's no longer in the blood vessel, it's now outside the blood vessel, but still outside of cells. It now has a new name. It's interstitial fluid. You can also call it ground substance. That's a term we'll use for it in connective tissues. So you'll see that very shortly. Now eventually, because this is happening all the time, we're constantly pushing fluid across capillary walls from the blood side to the interstitial fluid side. Eventually your blood volume would fall and your tissue fluid volume, your interstitial fluid volume would increase. And you would see that as swelling, edema. In fact, that's what's happening when you see swelling. We are forming filtrate faster than we are putting it back into the bloodstream. So eventually, let's take the cells away, just to simplify. There's another type of vessel in the same tissue that's going to pick up this fluid. It's a lymphatic vessel. And now the name changes yet again. As soon as we move this interstitial fluid across the wall of the lymphatic capillary, it's now called lymph. And again, this is why we don't have to monitor all divisions of the ECF, just one, just blood, just plasma. Because whatever the issue is with the plasma, you're gonna have the same issue with your interstitial fluid, with your lymph, and every other fluid that comes from capillary filtrate. So we do this, push fluid across capillary walls to form cerebrospinal fluid. So you have these capillaries in these cavities in the brain. We're just pushing plasma across, it forms a filtrate, but now it has a new name because of where it is. It's in the ventricles of the brain. We call it cerebrospinal fluid. We have capillaries in your synovial joints. And when that fluid gets pushed across the capillary wall and it's now in that synovial joint cavity, we call it synovial fluid. We do it in the eye to create the humors of the eye, and so on and so on. Everything is derived from plasma. Ultimately, it's all capillary filtrate. The job, or one of the jobs of the lymphatic system, is to maintain blood volume and prevent swelling of our tissues. To make sure that as we continue to make filtrate, and thus interstitial fluid around the clock, we make sure it's not in excess, right? As quickly as we form it, we pick it up and we return it to the bloodstream, because that's where the lymphatic system ends. It actually dumps directly into veins. And so your plasma became interstitial fluid for a minute, but then the interstitial fluid got picked up by lymphatic vessels, so then it's lymph. But then the lymph gets returned to the blood vessels, so now, once again, it is plasma. Yeah. So again, what was the driving force for moving the fluid across the capillary wall?
Hydrostatic.
Hydrostatic pressure, but we have a name for this particular hydrostatic pressure.
Blood pressure.
Blood pressure. So what is the function of the heart? To generate hydrostatic pressure. And we use that hydrostatic pressure for two things. To move blood through blood vessels, to pump blood, right, through the circulatory system, and filtration. And the kidneys rely heavily on filtration. That's how they do their job. We temporarily take the fluid that's in our blood vessels and we move it into a system of tubules in the kidney. We make a filtrate with it and run it through the system of tubules. And anything that we want to keep in the body, we're going to pull back out of those tubules. Anything that we need to get rid of, excess ions, waste products, even excess water, right? Maybe you consumed way too much beverage, you got to pee out the excess so your blood volume doesn't go up. All of that will leave. It'll go through the system of tubules, eventually out the ureters, bladder, it's urine at that point. But anything we want to hang on to, like all the nutrients, all the glucose, right? We pull it back out. So we have tons of transporters along those kidney tubules that are performing both passive and active transport, pulling out stuff that we want to hang on to that we don't want to lose. And see, that's exactly what I mean about this chapter 3 stuff, this membrane transport. It doesn't go away. To understand what the kidneys do, you have to understand membrane transport. That's all that it really is in the end. That's what they're doing.
Question.
Yeah.
I have a note that says filtration. Is that connected at all to?
The filtration that we were just talking about, that filtration.
There it is. Okay, passive. Yes, thank you. So that's all passive. Okay.
We good on filtration and osmosis. When filtration is occurring, what exactly is moving across the capillary wall? I mean, I know it's, the fluid is called plasma, but the composition, like what is actually getting across? Water, of course. Is that it though? That's what I'm getting at. What else is getting across, getting pushed across? All the dissolved solutes. anything that's in the plasma that can actually fit through the little gaps between simple squamous epithelial cells. Right? That's what the capillary wall is. And we have these little intercellular clefts, these little gaps between the cells, and that's how the water gets through, as well as everything that's dissolved in it that's smaller than what? Because what's too big to get through? Right, we had a little slide of this. Blood cells way too big to get through. Yeah, that's out of the question. So no cells are getting through here. I'm not talking about blood cells. I'm talking about something in the plasma. And it's the reason plasma is a colloid and not a solution. Proteins. Proteins don't get through. So that filtrate is plasma minus the proteins. Cells don't get through, the proteins don't get through, everything else does. That was called bulk flow. You remember the image? There's a slide that shows this, has the arrows and everything. And when we went over that image, I reminded you that this means there's now two ways for solutes to get into the tissues and to the cells that need them, say like glucose. Right? We need to get this glucose across the wall, through the interstitial fluid, across the cell membrane and into the cell, so that it can be used through glycolysis to make ATP. One way that glucose is getting across the capillary wall is by taking a ride with the fluid, the water that's moving across, right, through filtration. And that's why the term is bulk flow. Bulk is all the solutes that are dissolved in that water. It's not just water. It's all the other stuff too, smaller than the proteins. All right, so what's the other way glucose is going to get from the blood to the cell? Diffusion. So there's more glucose in the blood than the interstitial fluid. There's more glucose in the interstitial fluid than inside the cell. So you have a gradient to go from the blood all the way to the inside of the cell. Now, it's water soluble, so how does it get across the cell membrane? All right, so I'm doing that thing again where I'm just making connections all over the place to other things you need to know. What proteins? Yeah. With membrane transporters, right? Those are proteins. More specifically, how are we getting the glucose across the cell membrane? But we don't need active. We just said there's a concentration gradient for it to go in. So that information was important. Higher in the blood than the interstitial fluid, higher in the interstitial fluid than the cell. There's a gradient for diffusion, but it's hydrophilic, so how does it get across the cell membrane? Glucose is bigger than water. Carrier, mediated, facilitated diffusion. How about ions? How are they getting across the cell membrane? Channel, mediated, facilitated diffusion. Water. Channel, mediated, facilitated diffusion. What are the names of those water channels? Aquaporins. Translates to water channel. Next. Membrane receptors. Don't need to know too much yet. Structurally, what are they? So if you picked up on the pattern when I asked that, I'm asking what type of biomolecule is it? Membrane receptors are proteins. What are they used for? Receptors receive, right? What are they receiving? Chemical signals. Chemical signals. Biological markers. Like an immune cell is going to make contact with other cells in the body, looking for whether or not that cell belongs or is foreign. And if it belongs, is it healthy or is it infected? And this is a next semester topic, immune system. You'll see that when the cell is infected or mutated, it starts displaying abnormal proteins on the surface of its cell. The immune cell comes along and sees these abnormal proteins and says, you don't belong, and so I'm going to destroy you. Treats this abnormal body cell just like it would some foreign cell. For the same reason, it's potentially dangerous. And these abnormal body cells that have the mutations, I'm talking about cancer cells, tumor cells. All of us develop tumor cells, cancer cells in our life. But we don't necessarily get cancer because our immune system takes care of it, right? That means our immune system was successful at doing its job. Monitoring the body for foreign cells and abnormal body cells, find them and destroy them so they don't cause a problem. If you develop cancer, it's because your immune system got overwhelmed for whatever reason. The other example I think I gave you was blood transfusion. Why we do blood typing. It's because of those biological markers, those little flags on the surface of the cell. And if you're not receiving the right blood type that's similar enough to your own red blood cells, then those little flags on the surface tell your immune system this doesn't belong. And then you start destroying those cells that you just received through the blood transfusion. And then you have a transfusion reaction. It can be life-threatening. It can destroy your kidneys, for one thing. The other type of reception with receptors is really just receiving chemical signals, binding to chemical messengers, hormones, paracrine factors, neurotransmitters. Right, so when the neuron releases the neurotransmitter at a neuromuscular junction, so it's communicating with a muscle cell. The neurotransmitter binds to receptors on the muscle cell membrane. And what does that trigger? This would be a good time to bring this slide up now. Once the chemical messenger binds to the receptor, what happens next? And there's a few possibilities. This is chapter 3, I believe. Yeah, right after resting membrane potential, the next major topic, membrane receptors, provide sensitivity to the external environment. Kevin, when's the pizza coming? You ordered pizza, right? No? This is the least amount of food I've ever seen at a review. Usually like the whole back table is like potluck, bringing all kinds of stuff. And then they try to set it all home with me, like donuts and pastry. Can't do that. Well, when we do reviews in this room, you can bring whatever. It's just the other classroom, we can't have anything. So this cell-to-cell recognition on the bottom right, that was the example of the immune cell making contact with another cell in the body, trying to figure out if it belongs or if it doesn't, or if it's normal or abnormal. So this is the exact scenario I was just referring to. Neuron communicating with a muscle cell at a synapse called the neuromuscular junction, NMJ, which is the name of this synapse. The neurotransmitter is acetylcholine, binds to receptors. Those receptors are proteins, but those proteins are functioning as more than just a receptor. What else are they functioning as? You see arrows? That implies something's going through. It's a channel. So this one integral protein, transmembrane integral protein, is functioning as a receptor and a channel. And this is what you would call a ligand-gated channel. What's the ligand? The chemical. The chemical is the ligand. So you could just as well call them chemically gated channels. Ligand-gated or chemically gated. Traditionally, we go with ligand-gated. All right, so the chemical binds to the receptor, the channel opens. Looks like something's allowed to come in, something's going out. So I'll give you a hint. Those are ions. One is sodium, one is potassium. Is sodium the yellow one or the green one? Like these little. Sodium must be the yellow one because we see that the yellow one is coming right into the cell without help. This is just a channel. It must be going down its gradient. That can't be potassium. This must be potassium. Right? Because going out is following its concentration gradient. As ions flow across a cell membrane, what is created? Flow of ions. Electricity. Yeah, this is kinetic energy. It's electricity. We can measure it as a current. And what's going to be the consequence of this electrical signal that's being generated as the ions move across the membrane? What's it going to trigger? It's a muscle cell. Muscle cells do one thing, contract. Yeah, it's going to trigger contraction. So we're going to expand on this in chapter. Picture on the right. This would more likely be, it could be a neurotransmitter, but this is typically what hormones do. When hormones bind to receptors, it's usually not a ligand-gated ion channel. It's usually the first of many proteins in a cascade of reactions that result in the formation of an intracellular chemical messenger called the second messenger. Because this gets kicked off by the first chemical messenger on the outside, the hormone. Could be the neurotransmitter, could even be a paracrine factor. They're all capable of stimulating a pathway like this. And then what the pathway as a whole, what is that called? It's on your study guide. It's underlined. That means you better be able to identify it. That's what the stars represent. Things you need to be able to identify. This is a second messenger pathway. So binding to the receptor activates a G protein, which switches on an enzyme called adenylate cyclase, which takes ATP, converts it into cyclic AMP, which reaches kinases, switches them on. Those kinases then phosphorylate proteins in the cell. The proteins that get phosphorylated produce the cellular response, whatever it happens to be.
Like negative.
Nope. So you'll need to know this in detail next semester, endocrine system chapter. For now, I want you to know that there is this thing called a second messenger pathway. It is one possibility of what happens once the chemical binds to its receptor. Here's another possible action over here. It just opens a ligand-gated channel. This is very quick, very direct. It is in fact called a direct mechanism of action, because as soon as you bind to the receptor, the response occurs. The response is the electrical signal being created. Here, as soon as you bind to the receptor, there's no response. It's going to take a little bit. This has to activate that, has to activate that. We gotta make this, it's gotta activate that. We gotta phosphorylate proteins. This is an indirect mechanism of action. It takes longer to get a response. However, you get a huge response because that one chemical messenger binding to that one receptor can result in the phosphorylation of millions of proteins. Because once you switch on adenylate cyclase, you're going to be cranking out the second messenger. Right? So this pathway provides amplification. That original signal, right, becomes strengthened down this pathway. Here, one neurotransmitter opens one channel, and that's it. To create this electrical event, we're going to need to open thousands of channels, which means we're going to have to release a lot of first messenger, a lot of neurotransmitter, right, to get a big response. So faster, but smaller response. Slower, but bigger response. You get amplification. That's where we're gonna go with it next semester. So over here, what's the star? Just the chemical messenger, neurotransmitter in this case. This one, the ligand-gated ion channel. Here, first messenger here, second messenger here, the membrane receptor, right? Just the basics for now. Next topic.
It's just a question for the types of proteins. Do we need to know also the examples, like what kind? For the
Can you give me an example of the
like
example?
the collagen and
Oh, collagen, keratin?
Yeah, we need
You
to
don't
know
need to know
all
the
of them.
names of
Or
collagen and
just the
keratin.
basics.
But from the picture, can you tell me what level of structure they have?
That's all.
That's the question. It's kind of like the chemistry slides. You don't need to tell me based on the illustration, and that's glucose, and that's fructose, and that's galactose. You just need to tell me those are monosaccharides. So yeah, I don't expect you to be able to tell me what's keratin and what's hemoglobin, but just what they represent in terms of levels of structure. And what level of structure does keratin and collagen and hemoglobin have, and transthyretin? All examples that we looked at. Quaternary, right? They're all made of two or more separate proteins that have been joined together. So keratin, collagen, hemoglobin, those were examples of proteins with quaternary structure. Oh, you're talking about the levels of structure. Okay, so you have two basic types of proteins. We can call them fibrous proteins and globular proteins, or structural proteins and functional proteins, right? So globular and functional go together, and then fibrous and structural go together. To make a structural protein, you often stop at the secondary level of folding. You stop at secondary structure with your alpha helix, and now you have a nice long fibrous protein. It probably doesn't stop there. I don't know of any examples where we stop there. We're usually going to make a bunch of those and then connect those together to make a rope-like fibrous protein, and then that's where collagen, keratin comes from. So those proteins, in the end, have quaternary structure, but they did skip tertiary, right? We didn't have tertiary. We had 3 proteins, each with secondary structure, and then we connected them into one big protein with quaternary, skipping tertiary. Functional proteins, globular proteins, they all have tertiary, at least, right? And then we may take 2 or more proteins with tertiary structure and combine them into a big protein complex with quaternary structure. But it's still globular, still a functional protein. Hemoglobin would be the functional protein from those examples. Keratin, collagen would be the structural protein. Next. Right, so at home, you're going to go through all of these slides. You're going to make sure you know the story behind the slide. The title at the top is a huge hint. And sometimes there's not much to the story, and sometimes you can actually get into quite a bit of detail. So here, what's going on? Ideally, I make an unlabeled version of all the slides to actually do this, because now you can just look at the title, but it's fine. So this was to show what? That there are many different types of cells in the body. They look very different. They have very different functions. However, what makes them all the same, in a way? What is the thing that they all have that's identical? They all have the same DNA. And so how is it possible to have all these different cells, yet they have the exact same DNA?
They choose different genes.
Right. No cell has all of its genes active. So the set of genes, this is a specific combination or set of genes that become active in the cell, which in turn make a specific set of proteins. That determines the structure and in turn the function. So that's how they turned out so differently. They just activated different genes, made different proteins. What does a gene code for? Typically, one protein. One gene, one protein. What do you need to know about the generalized human cell model? Because this isn't any particular cell in the body. The models we have in the classroom, we can't say, oh, that's an epithelial cell, or that's this, that's that. It's a composite. It's like, here are all the most common features. When you look across all the different types of cells in the body, here are all the common features. That's what this is.
But not all the features, right?
Not necessarily, no. Like, there's no myofibrils, which is an organelle. They're not shown here, but we would find that in a muscle cell. But that's very unique to that one cell type. These are things that most cells have, almost all. Do you know of a cell that does not have a nucleus?
Is that the red blood cell?
That's right, red blood cells do not have a nucleus. Therefore, what can they not do?
Replicate.
Cannot replicate, no mitosis. What else?
They cannot carry any genetic information. Right?
Well, that's the given part. Now we want to know the consequence. Yeah, if you don't have a nucleus, there's no genes, there's no DNA. What are the two consequences? One, you can't divide your nucleus because you don't have one, so no mitosis. Two, what can't you do without DNA? What? Renew? Yeah, renew what? And it's the reason these cells only last a couple months, then they die and we have to replace them. What is it that you need to renew all the time because they break down? Proteins. What is DNA the code for? Proteins. Yeah. Proteins don't last forever. Once they're made, they last months, weeks. If someone gives you a shot of antibodies, those are proteins. They don't provide you protection for life. You're protected for a month or two and that's it. Because they fall apart.
Can I ask a related but off-topic question? So when somebody's bit by a spider or snake, right, that venom is proteins. But that's just a rapid decay
Can be
of the
proteins.
cells.
Say again, rapid
It's
decay.
replicating the cells. So instead of the cells regenerating, right, basically dying and then, well, not dying, it's a bad term.
Are you talking about the consequence of the venom?
Yeah, I'm just trying to get.
I think you made a huge jump. I
You know
didn't
what?
follow.
We'll talk later.
Okay.
I don't know how to phrase it
Okay.
in front of so many people.
I sometimes have that issue.
Performance anxiety.
You never believe I'm an introvert. Well, you might, actually, but total introvert. I couldn't do this in any other format. It's got to be A&P. It's got to be something I know well, I'm comfortable with, I enjoy talking about. But if you said, go up in front of 50 people and talk about this book you just read,
I don't have that problem. I'm just afraid of somebody stupid asking this question.
Well, that's part of it. Yeah, that's part of it. All right, so be able to identify, right, from pictures, from models, each of these organelles, and tell me very concisely, briefly, what's the function. Not to the level of detail in the book. Don't differentiate between microtubules, microfilaments, intermediate filaments. Just know that all of those things combined makes the cytoskeleton. Like are you talking about the pre-lab assignment? That's fine. Don't even worry about that. Yeah. It was probably a labeling activity. Was it part of the mastering assignment that I created, or was it follow-up? So it's probably like, well, 95% of it you need to know, so I'm gonna go ahead and give you the question, but that would be the 5% you don't need to know. The difference between those different fibrous proteins. Yeah, don't worry about it. Yeah, if I point to any fibrous proteins kind of crisscrossing through the cell, just say cytoskeleton. That's good enough. All right, so what's the function of the cytoskeleton? Do you need to know that? Provides the cell with structure, with shape. Yeah. And as you'll find out later, it does help transport materials within the cell. We have these little motor proteins that use the filaments and tubules like a track, and they slide up and down and move things around the cell that way. It's pretty cool. Smooth ER function. Synthesize lipids, detoxification, RER. Synthesize proteins that are either going to be
used as part of the membrane or
inserted into the membrane or
transported out.
Exported, secreted out of the cell. Because if you're going to make a protein that's going to stay in the cell, then you make it where? Free ribosomes. Yep. Don't need the RER. Once the RER makes the proteins, packages them in a vesicle, where does the vesicle from the RER go next? Goes to the backside of Golgi, which is called the backside. And then Golgi takes it in, maybe mixes it with some other proteins that were made earlier, maybe adds some polysaccharides, lipids, whatever, modifies those proteins, puts them in another vesicle, and this one's called a secretory vesicle. And then it's going to depart from the other side, the front side of the Golgi apparatus, and head towards what? And all of this can be seen. Cell membrane. And the membrane of the vesicle fuses with the cell membrane. And if the proteins we made are going to be inserted into the membrane, then they're not free floating inside the vesicle, they're actually already in the membrane of the vesicle. And then we just fuse the membrane of the vesicle with the cell membrane and done. If it's something that's going to be released, then they're not in the membrane of the vesicle, they're sitting inside. Right? And then exocytosis, right? That's what's being shown here. Function of nucleolus. Assembly of ribosomes, of the ribosomal subunits. That is correct. There's a lot you can discuss here. So rather than looking at the whole cell, we're focusing now on one of the three basic parts, the cell membrane. What can you tell me about the cell membrane? It's primarily a phospholipid bilayer, and then added to the phospholipids you have some glycoproteins, some glycolipids, and a bunch of other types of proteins, right? Glycoproteins are just one type, but lots of membrane proteins, transporters, enzymes, structural proteins, all sorts of stuff. What else? So we have phospholipids, glycolipids, proteins. Cholesterol is in there as well. if you have an area of cell membrane with a lot of cholesterol wedged between phospholipids, does that make it more fluid or more solid?
More solid.
More solid. Collectively, all of the sugars sticking out from the surface of the membrane, from the glycolipids and glycoproteins, that forms a covering called glycocalyx. So those carbohydrates, some of them individually act as biological markers for cell-to-cell recognition, but as a whole, collectively, you would say the glycocalyx does what? What function is provided by this? Sugar cover.
I know it makes it sticky.
Is it signal?
Cell-to-cell junction, right? It's sticky and it helps hold the cells together, but it's definitely not a strong junction. If you want to make sure those cells cannot be pulled apart, what are you going to use to connect them?
Desmosomes.
Desmosomes, right? A specific type of membrane junction. We know that epithelial cells, when organized into a sheet, are typically forming a membrane, a barrier of some kind, where we don't want just anything to cross. So if you want to make it difficult for substances to squeeze between those epithelial cells, what membrane junction do you use to join them? Tight junctions. What are they also known as? And the other name reflects the function. Occluding junctions. They prevent things from getting through. What was the nickname for desmosomes? Also reflecting the function. Anchoring, right? A strong anchor holding the cells together. Anchoring junction. Cardiomyocytes. When one of them becomes excited, when one of them generates an action potential, an electrical signal, it's going to spread throughout all the cells within a split second. How? How are we passing this electrical signal from cell to cell to cell to cell so quickly so they all contract at the same time? Gap junctions. So the flow of ions that are causing one cell to become excited flows through the gap junctions and excites the next cell, the next cell, the next cell. What are the names of the proteins within the gap junction? The gap junction is an area, but then it's studded with proteins, connexins. Those are the hollow tubes that allow the ions to pass through. Which lipids are amphipathic? Oh, don't mind me, I'm just waiting for your answer. What was the question?
Which lipids are amphipathic?
Which lipids are amphipathic? Phospholipids? Because they have two things that's required to be amphipathic.
Polar head.
They have an area, a region that is polar, the head, as we call it, and the region that is non-polar, which is the tail. Structurally, what type of lipid is a phospholipid, and for that matter, a glycolipid? Think about the structure, what it looks like. All right, how about the structure of a triglyceride? A glycerol backbone with 3 fatty acids, and the fact that it has 3 is why we call it a triglyceride. Phospholipids, glycolipids don't have 3 fatty acids. They are, and so their name, what are they? Diglycerides. They are diglycerides. What lipid consists of 4 interlinked hydrocarbon rings? Steroid. Most common steroid in your body. Cholesterol. What do we make from cholesterol? You think of cholesterol as being bad, you have to have cholesterol. Cholesterol has very important functions in your body. We make steroid hormones. All steroid hormones are made from cholesterol. And they structurally look very similar to cholesterol, right? Just modify the functional groups. It's incorporated into bile. It's important for digestion of lipids. How about a structural function? Contributes to the structure of the cell membrane. Yeah. Has some very important roles. Is the steroid lipophilic, hydrophilic, or amphipathic? Just lipophilic, not hydrophilic at all. All right, questions version, chapter 1. What are the 7 necessary life functions?
Maintain boundaries.
What was that? Missed it. Reproduction. Okay, reproduction. Responsiveness. Excretion. Yep. Okay. Movement. Yeah. Ventilation. Moving air in and out of the lungs. Right? That is the key function of the respiratory system. To accomplish the goal of gas exchange, you have to ventilate. You got to bring fresh gases into the lungs, into those little air sacs, to replenish the oxygen that is leaving the air sacs and going into the blood, right? And then you also have to push the air back out, clear out those little air sacs, because they're constantly picking up CO2 from the blood, and we got to get that out. Otherwise, we lose our concentration gradient. If you want to make sure that CO2 keeps going from the blood into the air sac, then you want to make sure that CO2 does not accumulate in those little air sacs. That's why you're pushing the air out during exhalation. If you want to make sure that oxygen is able to continually go from the air sac across the respiratory membrane into the blood, then you have to make sure that oxygen in the air sac is kept high. So how do we keep oxygen high and CO2 low? Ventilation. Ventilation. All right, so which of these 7 necessary life functions can you connect to the act of ventilation and the gas exchange that results?
Excretion.
What are you excreting?
CO2.
That's true, yes. So with what the respiratory system is doing, there's definitely an excretion component, certainly movement. How do you move the air in and out?
Diaphragm.
Yeah, by contracting and relaxing skeletal muscles. The 2 most important ones, the diaphragm, which is number 1, and then next to that, external intercostal muscles. All right, so think about the name, intercostal muscle. So it's a muscle found between, what's costo? The ribs. I heard responsiveness. Now, responsiveness is not something that the respiratory system is doing. That's something that another system is doing, which happens to be a control system and will in turn cause you to ventilate more or less.
Central nervous system.
The nervous system, yeah, is providing the responsiveness. But just, strictly for respiratory, we've got movement, we've got excretion.
What about boundaries?
Yeah. Yeah, the respiratory membrane is a boundary. What is it separating?
The in and out.
The internal environment with the blood on one side of the wall, and the external environment, what's actually inside the air sac, right? That's all part of the external environment. Besides actually breaking down materials from the external environment, which is to digest, that's to break down, what is the other key function of the digestive system? We are digesting for what purpose? It's a means to an end.
Nutrients.
We can't do this other thing without first digesting these raw materials. Absorption. Absorption. Right? So the digestive system provides digestion and in turn absorption of nutrients, of materials from the external environment. Which of these necessary life functions can you connect to the digestive system?
Excretion, movement.
Yes, so we bring things in to one side of this tube that runs through the body, and other things go out the other side, things that we don't need. Movement, all right, explain the movement. Peristaltic contractions. We actually move the foodstuff through this long tube, right, through the digestive tract. What tissue do we use to produce movement? It's one of the 4 primary tissues. We're doing that connecting thing again. Muscle. All right, what type of muscle? Smooth muscle. And when you use smooth muscle and you contract at one end, and that contraction as a wave passes down to the other and pushes the stuff in one direction, that's peristalsis, peristaltic contraction. Anything else? Clearly digestion for the digestive system.
Metabolism.
Yeah. What reactions are going on? Because metabolism simply refers to any reactions occurring and all reactions occurring in the body. Which chemical? They're all chemical reactions. Hydrolysis, right? Catabolic, those catabolic reactions, breaking down the proteins, complex carbohydrates into their building blocks. Yeah, it's all hydrolysis. But metabolism, absolutely. What do you notice after you eat a large meal, besides feeling sleepy? It has to do with metabolism increasing, the rates of these reactions increasing as you digest the meal. You get warm. It's the thermogenic effect of digestion. You get warm. As all those reactions speed up, as you process all of those raw materials. Is there a boundary involved in the digestive system? The mucous membrane, right, that lines the digestive tract acts as a boundary, separating the external environment, anything that's in the digestive tract, from the internal environment, what's on the other side of the epithelial lining of that tract. What does the mucous membrane consist of?
Water.
Two basic tissue types. I know all 4 are actually there, but what are the 2 major tissue types, making this as some say, the simplest of organs?
The water.
Tissue types.
All tissue.
What 2 tissue types?
Epithelium.
Epithelium. And what underlies every epithelium in the body? Connective tissue. What type? You know this now too. Areolar connective tissue. Yeah. So in the areolar connective tissue, rich blood supply. So you don't have to cross the entire mucous membrane. You have to cross the epithelium. And as soon as you do that, you're in that connective tissue. You're going to find your way into the blood. Besides moving things through the body, through these tracts, what other types of movement are required? What else do we need to be able to do? Again, necessary life functions. Just expanding on movement. You have to be able to move your body parts to manipulate objects in the external environment, and you need to be able to move your body. Locomotion. Okay. What is homeostasis? It's a state, it's a condition. So what you're doing is you're describing the state of the body at this time. You are maintaining relatively stable internal environment. And you can leave it at that, or you can say, despite a constantly changing external environment. Because if it didn't constantly change, we wouldn't need to respond to it. We wouldn't constantly change. But our internal environment will if we don't do something about it, if we don't prevent those changes. So what's the first step in preventing changes in those variables that have to be kept in a narrow range? You can't do anything about the problem if you can't detect it. Who does that? Who's detecting the problem? Who's monitoring the environment? The receptors, sensors. And if it's the nervous system, we do call them receptors. If it's the endocrine system, it's the endocrine cell, not a separate receptor. The endocrine cell itself is acting as the sensor and the control center. I can map that out. So my version looks different than the author of our book's version. So if the endocrine system is responsible for this, the endocrine cell takes care of these two things. Acts as the sensor, acts as the control center. If it's the nervous system, you have neurons in the brain or spinal cord in the central nervous system. And that group of neurons is acting as the control center. But they are receiving signals from other neurons who form the receptor. Right? So the neuron that forms the receptor is going to signal the neurons that form the control center. Two separate entities. But when it's the endocrine system, the endocrine cell takes care of both. And this only works if the control center has a built-in. The control center only knows if there's a problem if it has a set point. So we usually show that as a double arrow, meaning it's going to make a comparison between what it's supposed to be and what it currently is to determine if there's a problem that needs to be fixed. And if there is, the control center communicates with the effectors. Right? And it's going to be effectors, multiple. So the example that we spent the most time with, thermoregulation. Starting with thermoregulation in your house, with your thermometer, your thermostat, your heater, your air conditioner, right? And seeing that the thermometer is the receptor, the thermostat's the control center, your AC, your heating unit, those are the effectors that fix the problem. In your body, what are the receptors called? So in your house, thermometer. In your body, thermoreceptors. All right, so when I have you illustrate negative feedback loops, I'm not looking for the basic components of any negative feedback loop, receptor, control center, effectors. I'll be asking for the names of these things for a specific negative feedback loop, like thermoregulation. Right? So for thermoregulation, in place of receptor, you're going to put thermoreceptor. What's the control center when it comes to thermoregulation in your body?
Temperature.
You can give me the brain. The brain's the control center, that works. You could get more specific. In the brain, it's the hypothalamus. So thermoreceptors are going to communicate with the hypothalamus, the brain. And in turn, if there's a problem, the hypothalamus is going to send signals to, I mean, you can put the word effectors, that's fine, but you need to tell me what they are. We can do a list. How many are there? How many effectors for thermoregulation? 3? So we're, instead of just putting muscles, be more specific. Which muscles? Just skeletal. Not smooth, not cardiac, just skeletal muscles. Sweat glands?
Blood vessels. Blood vessels.
Oh, I'm glad you put it that way. Blood vessels, you would get half credit.